Hello everybody, this is Jason from STEM Nerds. Thank you so much for being here in today's video. I promised we would go over Punnett squares. We would practice them. In a previous video, we talked about the basics of genetics. If you haven't seen that yet, check that video out. I will put it in the upper right-hand corner as well as in the description below because you need to watch that before you can understand what we're going to be doing here. And what we're doing here is we're practicing Punnett squares. We're looking at the probability of certain traits being passed down from parent to offspring. So I have a few practice questions for you. We're going to head over to the whiteboard because it's just easier to do it there than it would be to do it on screen here. But question number one, if you cross an individual that is homozygous dominant for yellow eyes with an individual that is homozygous recessive for pink eyes, what would be the likelihood or probability of producing offspring with pink eyes? And so let's go head over to the whiteboard and we'll take a closer look at this. All right, so in this first example, we have to set up a monohybrid cross. And we're looking at eye color, and we've got some strange colors here. But again, the purpose here is not necessarily the phenotype. It's understanding how to set this stuff up. And so we've got homozygous dominant, and that homozygous dominant is for yellow eyes. And we're crossing that with homozygous recessive for pink eyes. So how do we set it up? First things first. I always find it easiest to pick, I've mentioned this before, but we want to pick letters that represent the genotype for our phenotypes. And the letters should be easy to distinguish. And so in this particular case, let's just choose the letter R because uppercase R is a lot easier to distinguish from the lowercase. So what that means for us is first thing we're going to do is we're going to say that capital R, that is going to be our yellow eyes. Lowercase r, that's going to be for pink eyes. So that's the first thing. That's step number one. Step number two is after we've figured out our letters, now let's figure out the genotypes of both of the organisms that we're trying to cross. So we said we have one individual that is homozygous recessive, and that homozygous dominant. So we have homozygous dominant for yellow eyes. We're going to cross with homozygous recessive for pink eyes. And so that's our cross. This is individual number one. This is individual number two. Next thing we have to do is actually set up the Punnett square. Okay, so step one, identify what letters we're going to be using. Step two, identify the genotypes of each organism. All right, so we said homozygous dominant, homozygous recessive. Step three is actually setting up the Punnett square. And so what we're going to do is we'll just put first organism up here, and the second one will be over here. And so what we're doing here with the Punnett square is we're trying to determine all of the potential alleles or versions of a gene that an organism can basically donate to this process. Well, if you're homozygous dominant, homozygous recessive, you really only have one letter. And so now we've got our Punnett square. And we're going to figure out, okay, for the offspring, what is the genotype of the offspring? And all of them are going to be heterozygous. They all have to look like that. It's the only option. You've got homozygous dominant up here, homozygous recessive. Each of them can only provide one version of the allele. Homozygous dominant can only provide the dominant allele. Homozygous recessive can only provide the recessive allele. And therefore, all of our offspring are going to be heterozygous, which means all of our offspring are going to show the yellow phenotype or physical characteristic. They're all carriers of the recessive allele, but they will all show the dominant phenotype here. 100% chance of yellow, 0% chance of pink. All right, so hopefully that is starting to make sense. Now let's take a look at question number two. Again, this is a monohybrid cross. We're only looking at one characteristic. So again, we're using the same general foundation that we used in the last question, except now, suppose you cross the two heterozygous individuals from the previous example. Both have yellow eyes. What would be the probability of producing offspring with pink eyes? And so in other words, we need to look at what is the probability of producing that pink eye phenotype. Let's go see. All right, so our next question said, hey, you know what, now we've got these heterozygous individuals. Let's go ahead and let's cross those and see what the offspring look like. And so now what we're changing is really up here. We're going to change all this around. So let me erase all of this. This hasn't changed, right? We could still use the same letters, but what the individuals that we're crossing are both heterozygous, which means they're both showing the dominant phenotype right now, but they're both carriers of the recessive allele. Okay, so same thing. We've identified the letters, which we're not changing here. We've identified the genotypes of each organism. Next thing we have to do is set up our Punnett square, but now here it's a little bit different because each of our individuals can either donate a dominant or recessive allele. So now what's the offspring going to look like here? So we have R here, capital R here, lowercase, capital R here, lowercase, and then 2 lowercase. Now I want you to see something. What I've done here is I always put the capital letter first. The uppercase letter is always going first, regardless of whether it was at the top or the side. It's just going to help you tremendously organize what your probabilities are later. So we've talked about some rules. Always identify our letters. Then we identify the genotypes of the organisms that we're going to cross. We set up our Punnett square. It didn't matter in the first one, but it matters here. Always, 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 our capital letters are going to go first when we cross. Now, what that means for us, if we look at this, anytime the dominant allele is present, the dominant phenotype is going to be expressed. And so homozygous dominant or the heterozygous, in all 3 cases, we're going to have yellow eyes. It's only in the case where we have the 2 lower case R's, where we have homozygous recessive, that the pink eyes are going to show up. And so the probability of offspring, again, this is probability, not exactability, but the probability of having offspring with pink eyes is 25%, 75% chance, or 3 out of 4 chance that we're going to have yellow eyes in our offspring. All right, now that we've got the monohybrid crosses down, we've got to do something a little bit more complicated. And this is about as complicated as we get, at least in these videos, and that is called the dihybrid cross. And in this video, or in this example, I should say, we need to look at 2 characteristics simultaneously. And so here's our question, and we're going to take it to the whiteboard just like we did before. Suppose you want to cross 2 individuals, but are interested in 2 different traits, or characteristics. How would you set up a cross between 2 organisms that are both heterozygous for eye color and hair color? And then I give you some assumptions here. Assume eye color, blue is dominant over brown, and for hair color, orange is dominant over green. What is the probability of obtaining offspring with brown eyes and orange hair? Let's go head over to the whiteboard and we'll take a closer look. All right, so here we go. Hopefully you understood those monohybrid crosses, because we're going to get into something a little bit more challenging, and that is the dihybrid cross. We're looking at 2 characteristics at the same time. In the question I just read, we've got both eye color and hair color. We've got blue and brown for our eye color and orange and green for the hair color. So again, we're trying to pick letters that are going to be easy to distinguish. So how about for eye color, we're going to say capital B, since blue is going to be dominant here. Our capital B is going to represent blue. Lowercase b is going to represent brown. Again, this is a letter where the uppercase and lowercase are easy to distinguish. For hair color, well, we can go H for orange because that's dominant. Lowercase h for green because that is recessive. And so we've identified the letters we're going to use. Remember, that's step 1. Pick letters that are easy to distinguish. So step 2, we have to identify the genotype of the organisms that we're crossing. Now, in the question, it said that both of them are going to be heterozygous for both traits. So, okay, that means we've got an organism that is big B, little b. That's eye color, heterozygous for eye, heterozygous for hair. And we're crossing that with an organism that is also heterozygous for both. We'll just call this number 1. Call this organism number 2. Now, here's where the confusing part comes in. So we got to have our concentration brains on. You ready? All right, concentration hat is on. So let's just put organism 1 here. We'll put organism number 2 down here. What we're doing here, remember, we have to come up with all the potential allele combinations that organism 1 can donate to new offspring. And so we have to kind of think this through. They have the capability of donating a capital B, right? A dominant allele for eye color. they also have the capability of donating a dominant allele for hair color. With this, we're picking from each one, right? Eye and hair. So we got capital B, capital H. We could do capital B, so all the different combinations, capital B, lowercase h. We could have blue and green. We could have brown and orange, or we could have brown and green. So those are all the combinations that we could potentially have that organism number one could, all the alleles that they could donate. Now, because they're exactly the same, the combinations are exactly the same over here. So now we have to come up with, there's going to be 16 different combinations of alleles. If you remember from the last video, you probably, if you remember from the last video, we already know the ratios here. But let's go ahead and work this out because we're trying to work out a Punnett square. So now we got the capital B and H here and here. So we got two big B's, two capital H's. That means this organism would have blue eyes and orange hair, be homozygous dominant for both. So we're just going to go through, I'm going to kind of go through this a little bit quickly. And I'm going to fill in all the letters as I go. And so here's a big B up here, one over here. So we're just going to go through. And notice what I'm doing again. All the capitals for the B's and the H's come first because that's just going to make it a lot easier. We're trying to figure out the probabilities in a few moments to know what the phenotypes are. And so I did that first one, then I'm coming down here. I'm just going to go through this. And hopefully when you do this, your handwriting is a little bit better than mine. So I fully admit, I don't have the best handwriting in the world, unfortunately for all of us. So again, I just filled in all of these, and we're going to go through and do this one. And again, we're going to have 16 combinations because we have heterozygous. Oops, almost made a mistake there. And you'll notice I'm always keeping the B's before the H's. And always, if there is an opportunity for a uppercase, I'm going to put that first for the B's or the H's. Okay. Always a great idea as you do these, just kind of double check and make sure that everything makes sense as you're going through, that you didn't make any mistakes like I did right here. And so we correct these. That's supposed to be two uppercase H's, right? You go through this quickly, you can sometimes make a mistake. It happens. Don't worry about it. Just make sure that in the end, you've got everything you're supposed to have. And I'm just going to double check real fast, make sure I didn't make another mistake while I was talking and writing at the same time, because that would make it harder for you to learn. And so I believe are we in good shape? It looks like so far. Okay, so here we go. Now, the question. We want to see, okay, what is the likelihood? Let me go ahead and read off my screen over there. What is the probability of obtaining offspring with brown eyes? So we want brown eyes and orange hair. So now here's the next thing we have to do. I know we've got the ratios that we talked about in the previous video, but we have to identify all the potential allele combinations that give us brown eyes and orange hair. So because brown eyes are recessive, that means the individual has to be lowercase b's, right? They have to be homozygous recessive. They have to be in order to get brown hair. But, brown eyes. But if we want orange hair, we can be capital H, or we could do little b, and then heterozygous for orange hair. So those are the two combinations that we're looking for. Okay, so now let's take a look. Where are little b's at? Here we go. This would work right here. This will also work, and so will this. So if you remember from the previous video, I said when you have a heterozygous crossing for both organisms, you have a 9 to 3 to 3 to 1 ratio. 9 out of 16 are going to show, there's a 9 out of 16 probability that both organisms are going to be showing the dominant phenotype. There's a 3 out of 16 that the organism is going to have a dominant phenotype with a recessive, 3 out of 16 that is going to be dominant allele with recessive going the other direction, and 1 out of 16 that we're going to be homozygous recessive. That's this one right here for both. So we're 3 out of 16, but there's another example I want to show you that I think is super important. All right, so we're going to do one last question in this video because I really want to make sure we understand the principle of the dihybrid cross. And so again, we're using the same criteria that we had in the last question, except now we're using the same characteristics as previous examples. Suppose you cross is now between one heterozygous individual and the other is homozygous dominant for eye color and homozygous recessive for hair color. Looking for the probability of obtaining offspring with brown eyes and green hair. Let's go head over to the whiteboard and see what happens. Okay, so in this next example, we're still dealing with a dihybrid cross, but the reason this is so important is a lot of students do a lot of extra work when they're trying to figure this next question out, and the work doesn't need to be done quite the same way we just did it. When we just did that dihybrid cross, we had 2 heterozygous individuals, we had 16 different potential combinations. With this next question, we don't always need 16 squares here. So, okay, I'm going to read off the screen over here. So it says using the same characteristics as the previous example. So here we go. Characteristics, traits. Suppose your cross is now between one heterozygous individual, so heterozygous for both, and so that hasn't changed. So organism number 1, capital B, lowercase b, capital H, lowercase h. But the second organism involved in this cross is homozygous dominant for eye color. So homozygous dominant for eye color and homozygous recessive for hair color. Okay, so homozygous dominant for eye color, homozygous recessive for hair color. How do we set this up? Little clue, we do not need 16 combinations. Now up here, for organism number 1, everything is still the same. So you can have both capital B and capital H, lowercase b, capital H, capital B, lowercase h, or let's fix that up. Capital B, lowercase h, and then we have lowercase for both. And so those are the 4 potential combinations that we have here. But for this guy, how many potential combinations do we have? Just one. Capital B, lowercase h. That's it. There is no other combination, so we don't need 4 things down here. I see students do this all the time. It just creates a lot of extra work. If you're being assessed, it's wasting time on your tests, on your quizzes, and then you have to reduce all the probability anyway and have the chance of making a mistake there. So with this one, because we only have one combination for the second organism in this cross, guess what? We only have 4 boxes, and then we can just go through like we did in the previous example, fill in all of our potential combinations. And our question was, what is the probability of obtaining offspring with brown eyes? Okay, so we got brown eyes and green hair. Well, guess what? To get brown eyes and green hair, we have to be homozygous recessive for both eye color and hair. And so that's what we'd have to have, lowercase b's and lowercase h's. Well, guess what? Right here, we got nothing. So there is a 0% probability, given what we have up here, the genotypes for the organisms that we're crossing, that we can have offspring with brown eyes and green hair. All right, everyone, so that was some practice with both mono and dihybrid crosses, how to set them up. In the next video, we're going to be talking about sex-linked traits, and we'll look at how we set up Punnett squares for those as well, because they're a little bit different. Thanks so much for being here. Have a great week, everyone.